Hi, my name's Diana Cook, and I'm a final year psychology student at the University of Galway. I was awarded the Social Nursing and Allied Sciences Summer Studentship by the Irish Cancer Society. Under the supervision of Nicolette Warner and Professor Anne-Marie Groark, I investigated the impact of social support, anxiety, depression, and coping mechanisms on quality of life in individuals who have been identified with predispositions to cancer. I applied for the studentship as not only would it allow me to develop my own study and gain first-hand experience of the work of a researcher, but it would also help contribute and support the work of the Irish Cancer Society. My main personal goal for the studentship was to further my knowledge of the psycho-oncology research field, as I hope to work in this domain in the future. So just a bit of background in case anyone's not familiar. Hereditary cancer conditions are caused by alterations in genes that are passed down through the family tree. These alterated genes increase the risk of developing certain cancers throughout the lifetime, and it is estimated that approximately 5 to 10% of all cancer diagnoses come from hereditary cancer conditions. Identification of an increased risk of developing cancer is a life-changing event, and dealing with this can be an extremely difficult experience, causing people to have feelings of guilt at passing the gene along to their children and stress over knowing their new elevated risk and dealing with appointments, the financial costs, and the lack of guidance through the experience, which often results in symptoms of anxiety and depression impacting their quality of life. Additionally, people may develop maladaptive coping mechanisms, which further induce feelings of anxiety and depression. Previous research has found that those identified with hereditary cancer conditions often suffer from much higher levels of anxiety, depression than that of the general public. Social support through healthy communication with family, friends, peers, and in the workplace has been found to reduce anxiety and depression levels, suggesting that improvements in social support may improve quality of life. As highlighted as an unmet need in the Irish Cancer Society strategy, there is a deficit in readily available psychological services for those identified with predispositions. This, combined with the predicted exponential increase in those identified with hereditary cancer conditions due to improvements in testing and testing technology and more widespread genetic testing, indicates the importance of finding immediate methods to alleviate symptoms of anxiety and depression to reduce the burden on the already lacking services. The first step in this project was discussing the idea with a public patient involvement panel, or PPI panel. To me, the meeting with the panel was the most memorable part of this project. As this was a quantitative study, there were a lot of times when the project could begin to feel robotic and impersonal, but hearing real life experience and discussing the topics showed just how valuable and important this work was. The panel felt like a key motivator to me for this project. The panel was used to make sure that the research we did would not only be useful, but would actually represent their thoughts, feelings, and experience. The panel offered additional ideas on areas that we could explore for the project. And because of this, we added workplace support as an additional area to research, as it became very clear very quickly that it was an extremely important everyday topic that we had already originally overlooked. I created the survey through Qualtrics, a platform that collects data on demographic information, communication in family, support from the family, friends, peers, and in the workplace, coping strategies, and quality of life. With the help of the PPI panel, we shared the study through different hereditary cancer online community groups to recruit for the study. We collected information from 94 participants and were able to reach 9 different hereditary cancer condition types as seen in the pie chart. In the sample, we found that overall, when participants had low levels of anxiety and depression, they had much higher levels of social support and quality of life, while participants who had high levels of anxiety and depression had low levels of social support and quality of life. Additionally, this study found that participants favored problem and emotion-focused coping styles, which may have attributed to the overall low scores of anxiety and depression and higher levels of support in the sample. This research strengthens the theory that social support is a beneficial way to reduce symptoms of anxiety and depression that impact on quality and life in those identified with hereditary cancer conditions, and highlight the need for promotion of healthy communication within the social circle and government financial investment into the service. It should also be noted that overall, there was very little workplace support reported, and this is an area that should definitely be further investigated. And in the near future, I would like to elaborate on this study for my final year project. Finally, I would just like to say thank you to Nikki and Anne-Marie and the PPI panel for all the help that they've given me throughout this project. And thank you for listening to me today.
Hi everyone, thanks for letting me speak here today. So my name is Mark Lee. I was one of the translational summer students, and I'm just going to talk to you today about the project, so the science that underpins it, the method, so what we actually did during the project, and how I found the lab experience. So here's me. I'm a UCC student studying BSc Medical Health Sciences, so it's a course geared towards looking at both the clinical and the laboratory side of science. So it's essentially geared towards translational medicine. So since starting the course, I've been interested in the cancer side of things and drug development. So I really jumped at the opportunity to combine those two fields. So I approached Dr. Krajewska, my supervisor, and the project title that we came up with is Harnessing the Potential of mRNA Delivery for the Treatment of Triple-Negative Breast Cancer. So triple-negative breast cancer, for those who may not have heard of it, it's a very aggressive subtype of breast cancer, killing a third of patients. It's relatively common, accounting for up to 20% of breast cancers. And it's defined by the absence of these three proteins, so estrogen receptor, progesterone receptor, and HER2 receptor. So those proteins, they're very commonly found in other types of breast cancers. They basically stick into the surface of a cell and cause them to grow exponentially. But if you don't have any of these proteins, then you don't have any of the normal drug targets, which means that the majority of drugs that target breast cancer will not be effective for triple-negative breast cancer. So there's really a lack of available treatment options, and with this project, we just aim to possibly generate more treatment options for these patients. So messenger RNA, you've probably heard about it before from the Pfizer vaccine for COVID-19. It's essentially middleman between DNA and protein. So we need our proteins to do basically every single function. But since we can't really change the DNA, what we can do is put mRNA into a cell, and then it will make the protein for us. So here's a diagram of most breast cancers on the left and triple-negative on the right. So as you can see, those proteins I mentioned, they're all stuck into the membrane or the surface of these normal breast cancers, and they're not present in triple-negative, which means that if you were to add in most conventional cancer therapeutics for breast cancer, they would stick onto these proteins, but since the proteins aren't present in triple-negative, then it has essentially no way of killing a triple-negative breast cancer cell. So for our project, we thought that if we could use mRNA encapsulated in a lipid particle, that's just the delivery system, that the mRNA would insert into the cell, and then the cell would generate these therapeutic targets, the proteins that wouldn't have otherwise been present, which means that then if you were to go adding a conventional therapeutic that would have been used to treat another type of breast cancer, it's able to stick onto the triple-negative breast cancer cells and kill them. So that would open up a wide range of therapeutic options for these patients and possibly improve the prognosis long term. So overall, the project objectives were to grow the cells, treat them with mRNA and see if it increased the amount of proteins, and then see if the drugs are more effective in combination with this mRNA. So here's two nice little pictures of the cell types that we grew. The triple-negative breast cancer in the control group, these kind of cancers which would have those three proteins. So you can see there's a big difference between them, just purely because of the presence of these three proteins. So the triple-negative breast cancer cells, they grew actually, I'd say, almost twice as quickly as the other cancer types. So you can see why it's quite aggressive. So here are the results. I'll just talk a little bit about them. So on the bottom left, that's called a Western blot. It's just looking at how much protein is in a group of cells. So down here, you can see that the mRNA caused a big increase in the amount of those target proteins. So that's very promising. means that the mRNA is very functional. And then up on the right hand side, this is how many cells we were able to kill in combination with the drug and the mRNA. And you can see that the mRNA was able to increase the effectiveness of the drug in one of these cases, but not all. So the results are quite positive, and it allows scope for further research. So my overall experience was very, very positive. I learned a lot of stuff in the lab, just how to measure proteins, different therapeutic analyses, reporting results, but also soft skills. So communication skills by giving my weekly presentation to my supervisor, working as part of a team to avoid stepping on everyone's toes in the little lab. So independence, working by myself down in the cell culture room. And of course, since so many things tend to go wrong in the lab, critical thinking to overcome these problems. So in terms of how the studentship has shaped my future plans, I did really enjoy the laboratory environment, and I think I am well fit for a career in research. So I'm now applying for a PhD with Dr. Krzyzewska, my supervisor, in the hopes of continuing this project and hopefully getting a drug out of it at some stage. So just to say a few thank yous. Of course, I'd like to thank Dr. Krzyzewska for her mentorship and guidance the whole way through the project. I'd like to thank the Irish Cancer Society for supporting this project, and the School of Biochemistry in UCC for lending me all their facilities. Thanks for listening to me, and I hope this was beneficial.
Hello, my name is Sarah Finney. I'm a final year student from RCSI. My project was titled Physiotherapy for People with Advanced Cancer in Specialist Palliative Care. And what we wanted to see was everyone getting the opportunity to benefit. So just a little bit about myself. As I said, I'm in RCSI, final year student in physio. I'm based in Dublin. And my main motivation for this project was that just I have an interest in health and exercise. So for this year in college, I do have a palliative care module. So I wanted to get an introduction to palliative care, particularly in an advanced cancer setting, just because we do see that 75 to 80% of patients that we do see in palliative care are patients with advanced cancer. And there is that recognised underutilisation of physiotherapy for these patients, with low referral rates being kind of published across all areas of research that I've seen, even though there's papers upon papers of benefits for particularly breathlessness and mobility, but also that physio can affect fatigue and pain positively. So this led to this project. So we started with a retrospective case series. So what that means is we went back to healthcare records and pulled information from it, which helped shape our aims. So we wanted to explore overall referral rates to see if there was an improvement from a previous study that was done in St. Francis Hospice in 2018, and if there was an increase or a decrease, and if so, what characteristics these patients had, and if we could kind of pull that from it and present it to doctors and nurses to maybe increase the rate. So we started with 677 patients. The staff from St. Francis Hospice got that down to 375. And from that, I analysed 97 healthcare records, and of those 97, only 40 were referred to physio. So that comprised 41.24% referral rate, median age of 75.9 years. So that was kind of the middle age of the patients. There were a couple of outliers where one I think was 45 and then all the way up to 99. We had a pretty even split of male and female health care records, so 46 males, 51 females. And the most prevalent cancer diagnoses that we found were lung cancer with 24, and then colorectal and upper gastrointestinal were 11 each. So our main kind of focus was, were people being referred? So we looked at associations between different characteristics. And the only characteristic that we found was linked to referral to physio was bone metastasis. but ongoing treatment had no effect. Social history, so whether they lived alone, had a healthcare package, if they died at home, died in hospice, none of that had an effect on whether or not they were referred. And interestingly enough, disease phase also had no effect. So if they were stable, they weren't more likely to be referred, as opposed to those who may be unstable or deteriorating. Then just a bit on the actual service utilization itself. So median time to referral to physio was 25 days, which doesn't sound like a lot, but when people have their initial medical assessments, you would kind of want them to be referred on that day. And only 8 of those were referred the day of their initial assessment, and then 14 were referred within the first 30 days, with most of the patients then being referred after those 30 days. And what I found really interesting was the most common reason for referral was mobility. But if we move on from there, we can have a look at, I'll just jump to mobility and physio-specific symptoms. Only 39.7 patients with a mobility issue were actually referred. And the reason we split them up into physio-specific and non-physio-specific is that we can't really have an effect on non-physio-specific symptoms, and it would kind of help us understand maybe why patients weren't being referred, particularly for upper GI symptoms. So that's nausea, vomiting. Patients just aren't up for participating in exercise. It might just make them feel a bit more sick, which we don't want to do. But definitely what jumps out at me is that patients with mobility issues was the big one, and pain as well. Only 39% were referred. And even at that, like none of them were above 50%. So moving on to the next slide. So if we kind of jump between the two slides, if patients had 4 symptoms, they would have had to have had at least one physio-specific symptom, which is interesting because the majority of patients lie between the 4 and 7 symptoms, and only 41% were referred. So I thought that was quite interesting. So ultimately what this means is no, not everyone is getting the opportunity to benefit from physio with advanced cancer, which highlights the need for more research in this area so that we can take our evidence and bring it into practice. So just a little bit on my experience of the studentship. So I was involved in designing the project, but a lot of it was kind of based on a previous study. So my main role was to develop a data extraction tool for extracting the information from the healthcare records, and then from that input it into data Excel sheet. And I got introduced to Stata, which was a fun time because I had used SPSS in the past, so getting to use another analysis tool was a bit tricky, but we got there in the end. Also coming into final year, this will be important because I do have a thesis coming up, and I hope to use this project to maybe shape a new one. So scientific writing was one thing that I really improved. I also improved communication skills, so I got to work as part of a research team. So that was alongside Dr. Ailish Malone from RCSI, and then specialist palliative care physiotherapist Fiona Cahill, and physiotherapy manager Lisa McGurr from St. Francis Hospice. And I was really blessed that they let me bridge the gap between my academic research and also clinical work. So they allowed me to go onto the wards before my placement. So I kind of got an early introduction prior to starting, which was good. I got to know all the nurses, doctors, so it made that transition a lot easier. So from that experience, my attitudes have also changed. So I gained insight into working in a research setting, but also combining it with clinical work, which I would like to do, and I hopefully go on to a long career in physio. It introduced me to a palliative care environment, and I actually requested to be part of the ongoing research. So the next study that we plan to do is kind of take it to doctors and nurses of St. Francis Hospice, just to kind of understand maybe why patients aren't being referred, if there's beliefs or attitudes there, or if it's just purely lack of staff, lack of resources, that it's just being missed. But yeah, that's everything. Thank you.
I think Shani might be frozen, but we'll just give her a minute. Okay, so while we just wait for Shani to load up, I was going to give a little blurb anyway about the summer studentship. So I might just take this opportunity to talk through that now, and hopefully once Shani rejoins, we'll be good to go. So just to let people know that every year the Irish Cancer Society runs the call for summer studentships around December and January. And the objective is to offer kind of excellent undergraduate students the opportunity to undertake a cancer research project and to work with researchers in high quality environments. So it gives students the opportunity to gain experience at an early stage in their career path. So we usually encourage undergrads not in the final year of their degree to apply for that, and then maybe start thinking about potential projects and supervisors now. But I can see that Shanu has joined us again. So Shanu, work away.
Yeah, sorry about that. Okay. Okay, so my name is Shanu, and I'm going into my second year of graduate entry medicine at UCD, and I was working in Dr. Thorpe's lab this summer, creating a collagen-based hydrogel system to model pancreatic cancer. So a bit of background. So the most common type of pancreatic cancer is known as pancreatic ductal adenocarcinoma, and it's one of the deadliest cancers in the world due to its low survival rate. So usually within 5 years, there's a less than 5% chance that you'd end up surviving. And part of it is because the tumor is covered by a mesh-like barrier that's made out of collagen. And in healthy cells, basically collagen provides a lot of support and structure, but when it surrounds these tumors, it makes it difficult for drugs to be able to attack the cancer, as it wouldn't be able to reach the tumor. So my project is to create a material that mimics that collagen meshwork that you would see in these tumors using hydrogels. And hydrogels specifically, because they're mainly made of water, they're a great sort of system to use to sort of mimic the human body, as we're made of 70% of water. So the first part of this project is creating these collagen gels. So I basically made gels at different concentrations. So at 1 mg per mil, 2 mg per mil, and 3 mg per mil, just to see how the effect of concentration would have on cell behavior. And then I incorporated those cells into collagen gels. So I used 2 cell lines, metastatic and non-metastatic. And basically I was able to resuspend the cells into these gels, and then incubated them so the gel would form with the cells inside. And then I would assess the gels to see if the cells were able to survive the sort of encapsulation process over the course of multiple days, and as well as to see if the cells would be able to contract the gels. And this basically went on a week-long sort of time course. So the results regarding the cell viability just to see if they would survive, I used fluorescent dyes to see the proportion of live and dead cells. So basically, there's 2 dyes that I used. The calcine AM would cause the cells that are alive to fluoresce in a green color. And then the ethidium homodimer is used to cause dead cells to turn red. So I did this twice. So at day 1 after I made the gels, and then day 7. So you could see after day 1, regardless of concentration and cell type, all the cells are alive, which is always a good sign. And then by day 7, only less than 10% of them were dead for both of them. So overall, this means that these hydrogels have a high cell viability, as these cells, regardless of if they're metastatic or non-metastatic, they were able to survive being within these gels for a long period of time, which means it would be a good use, basically an accurate model to model pancreatic cancer. And the second part of my project was creating this sort of gel contraction assay. So basically, I took photos of these gels every day using my iPhone camera, and then comparing how much the gels contract based on concentration. So on the left here, we have the metastatic cancer line, and on the right is the non-metastatic. So you can see at the lowest collagen concentration, you'd see the highest amount of contractility. while for the highest concentration of 3 mg per ml, there's not much contraction. And this is sort of what you would expect, because when you look at this assay done on different like other cancer lines, you'd see that lower concentrations cause higher contractility, and that's because the gels are softer, so it's easier for the cells to sort of get them to contract. And basically, the significance of this is that this means that this model could be used to sort of study cellular contractility as well as tissue remodeling in future studies. So overall, my summer studentship experience was really great. I obtained a variety of skills because basically I did an undergrad in chemistry, so I wasn't really exposed to any sort of biology stuff. So it was really neat being able to work sort of in cell culture and learn more about cell staining, as well as working in a sterile fume hood, like in the tissue culture lab, and learn the various sort of hygiene techniques sort of that come with it, because nobody wants an infection. And also I learned a variety of analytical skills in statistics, specifically when it came to analyzing my data to see if any of the results I've obtained over the course of the 8 weeks were significant. And I also gained a variety of soft skills such as communication, teamwork, and collaboration, as when it came to problem solving and troubleshooting my project, I would often turn to other members of my lab for help. And I also did a variety of oral presentations throughout my studentship, which really helped me become a better public speaker. But yeah, so that's my whole project. Thank you.
So brilliant. So thank you so much to our 4 outstanding speakers. And again, we welcome some questions in the Q&A box or in the comments. So just before Shanu started to speak, I was just highlighting about our summer studentship. So again, we would really encourage any students not in the final year of their undergraduate degree to apply for this and maybe start thinking about projects. But alongside that, it's a really good opportunity for early career researchers or academics to get some hand-on experience with supervising funded research projects. So I might start with a question, and it's more so just kind of getting a better idea of how you found the summer studentship. So maybe starting with Diana, since you presented first. Diana, you spoke a little bit about the PPI and how much you enjoyed that, but maybe you might want to tell us a little bit maybe about what you found frustrating about research and how you felt, how you kind of overcame that. So you talked about the highlight being PPI, but maybe what was the more frustrating part of the research process?
I'd say the hardest part about the project also came from the PPI when they highlighted workplace as like an important area to them. As soon as we started researching into it, it became evident really, really quickly that this was an area that just had very little research on it. So trying to find anything we could use for our study made it virtually impossible to get something that we could apply to this. But because it had been highlighted as such an important aspect of the project, it was something we really wanted to include. So we struggled our way through and eventually found a scale to use. And what we found was really beneficial in that it's a really problem area that really needs research invested into it.
Thank you. I might pass that question on to everyone in the panel because I think that might be an interesting learning. So after Diana, we had Mark.
Easy one now. For anyone who's heard of what's called a Western blot, it's just for quantifying the protein. It's basically like a 5-day long experiment. And if anything goes wrong along the way, you don't notice until the very last stage when you get your results up on the screen. So we had a few hours spending the whole week grinding, trying to get that done. And then Friday afternoon, I'm looking at a blank computer screen with no results on it. So yeah.
Okay, so you probably learned a lot of problem solving skills.
Oh.
Great. We have Sarah.
I would probably say the actual data extraction. It was just very tedious going through each healthcare record and just trying to pull everything. And a lot of the time, the records were missing information. So obviously you can't make that up from the past. So that we had to kind of increase our inclusion and exclusion criteria just to make sure we had everything. But yeah, got there in the end.
Brilliant. Okay, and then Shanu.
I guess it's just planning for experiments and assuming it's going to go a specific way, and then it goes the total opposite way. So that was pretty frustrating, but I guess because I was able to ask people in my lab for help and stuff, it became less frustrating. But yeah, things never go to plan.
I think that's a big learning in research. We have a question for Sarah. So Sarah, can you talk a little bit more about the number of people referred with brain mets and perhaps why this was the highest number of referrals? Do you think the staff required more education on the role of exercise and palliative care to encourage referrals?
So it was actually bone mets that were the most highly referred, but brain mets were definitely, actually we only had 2 healthcare records with brain mets, and they actually were both referred. I think that's because there is that recognised need for more neurology-based physio and mobility. So that was actually good to see that they were being referred. I suppose bone mets were referred, which was different from the study in 2018. Because research had come out to say that resistance exercise was actually okay for patients with bone mets, just to stay away from kind of high impact, because you obviously don't want any pathological fractures. I suppose when you're talking about education of staff, I think that's what our next study is going to be. So we were hoping that there would be a clear trend as to what characteristics and referral patterns would emerge, to take it to the staff and say, okay, these are the ones that are being referred, these are the ones that aren't being referred, but there actually wasn't really a clear trend at all. So now that's why we need to go to the staff, do a survey and understand why they're not referring, to maybe then gear an education or presentation on what we can actually do for patients with advanced cancer in palliative care.
Yeah, and that's a great finding to know that you need to go back to the staff and kind of dig around a bit further. Will that be a qualitative study that you're planning?
Yes, it will be. Yeah.
Cool. I have a question for Mark just as well with regards, you said you were planning on applying for a PhD. Are you going to take the findings from the summer studentship and develop them further for your PhD, or will you continue in a different branch?
No, it'll be the same branch. I'm hoping to do my FYP in this as well. So I'll just kind of keep it going as long as I can.
We have a nice question for everyone, which is what was the best part of your summer studentship research? So maybe Shanu, do you want to go first? We'll go backwards now.
I think my favorite part was working in a biology lab rather than a chemistry one, just because I don't know, I've never done cell work before, so it was very interesting just to see my progress from knowing nothing about cells or any how to basically the protocols that go with working in a biolab, and now being pretty proficient in a bunch of different skills that I wouldn't have thought I would have ever learned.
Brilliant. Sarah, do you want to talk about the best part?
I think it was the crossover between research and clinical work. So obviously I would have been reading a good bit about kind of the benefits of physio, but actually being able to see it on the ward and the patients are actually enjoying it, being able to get out of the room, do a little bit, maybe do a little bit more than they did the day before, or even patients coming in for classes, just having that social interaction was nice to see. So it's not all just kind of on screen, you're actually seeing the benefits and what your research could actually kind of help with as well.
That real life benefit to research. Brilliant. Diana, do you want to hop in?
I think one of the best parts was getting to talk to different people who responded to the study and getting to hear their stories and knowing how important the research was to them and what an impact this will actually have on their everyday life. It was just really nice to know that you were doing something worthwhile.
Yeah, absolutely. So a little similar there to Sarah, it's just about that real life impact of research. And then Mark, do you want to?
Yeah, I think I just got a thrill really out of seeing positive results. Because you can go through so many frustrating experiments and then you optimize and then eventually all of a sudden it just works. There's no better feeling really.
Brilliant. Okay, so I think we've asked you all questions and you did brilliantly and they were really interesting talks. So once again, just a huge thank you for all our attendees, and a huge thanks to our 4 brilliant speakers for showcasing the great work that you did over the summer. I'm sure it was a lot of hard work and effort went into it, and it's great to see that you seem to enjoy the whole process. So from us at the Irish Cancer Society Research Department, we just want to say a huge thank you to our summer students, and again would encourage everyone maybe who's listening in kind of the end of the year to look and see if they'd like to apply. So I think we might call it there. I think that's pretty much all of our questions. Oh, actually, Diana, there's a question in for you, and then we'll finish on that if that's okay. It's from Roberta. So after hearing Diana's positive experience incorporating PPI into your research, would it encourage any of the rest of you to consider doing the same in future projects? So maybe it's not a question for Diana, but more so a question to the other 3, and then we'll call it there. So I don't know, Sarah, do you want to start? Have you thought about PPI for future work? It might be something for a bigger project.
I haven't really thought about it. I suppose maybe it'd be worth looking at it in the future for something bigger to pull it in to maybe get a broader look at it. But I think for now we're very set on the qualitative survey of staff, and then
Absolutely. It might be interesting to include staff in the analysis who didn't take part in the research to see how they feel about the way results were analysed. I know in translational research, sometimes there can be different barriers, but Mark, do you have any thoughts on including public and patients in your research design development process?
Yeah, no, I think it's really useful just for kind of tailoring your research so you don't go off in the wrong direction. I'd say like down the line, if this research leads to maybe a therapeutic, or we could possibly ask patients and public what kind of delivery method might be most suitable, and see how we can kind of tailor that towards people actually wanting to take it.
Yeah, brilliant. That's excellent. And Shanu?
Yeah, I think it would be interesting because I'm hoping in the future I can get involved in public health and clinical trials. So it would be something I would be interested in doing.
Brilliant. So thanks to Roberta for that question. Diana, I think you might be slightly off the hook, given that it was after hearing Diana's positive experience, but would you have a final statement on whether kind of in the future you might look at doing PPI? Is there anything you do differently?
I think involving PPI panel was the best thing for the project, because it really honed in on what was important, and it just got the points across that we needed, that this was an important project, these are important points of the project and why they were important. And I think when you're just reading information, it's a lot different to hearing it from a real life person. So it emphasizes how important it is.
Yeah, brilliant. I think that's a lovely note to end on. So once again, thank you to everyone for attending. Thank you to our 4 fabulous speakers who did a great job today. And we have recorded the session, and just a big bullabus to everyone as well. So thank you.